Hey everyone, welcome back to another explainer. So today we're tackling a topic that honestly often feels like a bit of a biological grab bag. But don't worry, we're going to organize it so cleanly that you literally won't ever forget it. Consider this explainer your ultimate high-yield exam survival guide for Kingdom Protista. We're going to clearly separate the 5 major groups of this kingdom, and we'll be focusing heavily on those sneaky key classification differences that examiners absolutely love to test. By the time we wrap up, you'll have a crystal clear mental framework for all these organisms. Let's get into it. All right, let's quickly run through our syllabus for today. First, we'll meet Kingdom Protista. Then we'll look at the photosynthetic producers, followed by the shape-shifting euglenoids and slime molds, the decomposers. After that, we'll break down the 4 types of protozoans, and finally wrap everything up with our key exam takeaways. Section 1. Meet Kingdom Protista, the single-celled bridge. Okay, so to absolutely crush your exam, we first need to establish a solid baseline. These are primarily aquatic organisms, and they form a super vital evolutionary link between plants, animals, and fungi. Now, the strict textbook definition of a protist includes all single-celled eukaryotes. To be fair, biologists will be the first to admit that the exact boundaries of this kingdom aren't perfectly defined. I mean, what one biologist calls a photosynthetic protistan, another might argue is just a plant. But for your exams, the absolute constant rule is their eukaryotic cellular structure. That means their cell body contains a well-defined nucleus and other membrane-bound organelles. They might also have flagella or cilia to move around, and they reproduce both asexually and sexually, which involves cell fusion and zygote formation. Moving on to Section 2, the photosynthetic producers, specifically chrysophytes and dinoflagellates. Both of these groups are famous for producing energy through photosynthesis. But here's the kicker, they're equipped with drastically different cellular armors. And I want to heavily emphasize this right now, because it is an absolutely critical contrast for your exams. When we talk about chrysophytes, which includes your diatoms and golden algae, they feature cell walls that form 2 thin, overlapping shells. Think of it like a microscopic soapbox. And those walls, they're embedded with silica, making them literally indestructible. On the flip side, we have the dinoflagellates. These are mostly marine organisms, and instead of silica, they wear these stiff cellulose plates right on their outer surface. They usually have 2 flagella that sit in a little furrow between those wall plates. So lock this comparison in your memory. Silica soapboxes for chrysophytes, stiff cellulose plates for dinoflagellates. Now, because those silica walls of diatoms are indestructible, they leave behind some pretty massive real-world impacts. The accumulation of these shells over billions of years creates what we call diatomaceous earth. It's basically this gritty soil, and it is highly useful. In fact, this gritty, ancient accumulation actually filters the modern syrups and oils that you consume every day, and it's even used in polishing. Oh, and not to mention, diatoms are the chief producers in our oceans. They're an enormous deal. But hey, it's not all just filtering syrup. This brilliantly illustrates the really lethal real-world stakes of our other photosynthetic producer, the dinoflagellates. While they can be yellow, green, brown, or blue, it's the red dinoflagellates, like goniolax, that you really need to know about. When conditions are right, they undergo such rapid multiplication that they actually make the entire sea appear red. You've probably heard of it, red tides. And the toxins released by such an enormous number of these microscopic organisms, they're potent enough to wipe out huge numbers of other marine animals, including fishes. Section 3. The shape-shifting euglenoids. Ditching the cell wall. Found mostly in stagnant freshwater, these fascinating little organisms uniquely ditch the traditional cell wall entirely. They swap it out for something way more flexible. So the crucial point for your exam is this exact definition, the pellicle. Seriously, mentally circle this buzzword in your notes right now. Instead of a rigid cell wall like the cellulose or silica we just talked about, euglenoids have this protein-rich outer layer. This gives them a huge structural advantage, making their bodies incredibly flexible and maneuverable as they swim around using their 2 flagella, one short and one long. But you know what's really wild about euglenoids? It's their unique double life. When they're in the presence of sunlight, they act as photosynthetic autotrophs. And interestingly enough, they use pigments that are absolutely identical to the ones found in higher plants. But when they're deprived of sunlight and cast into darkness, they completely shift their behavior. They literally become heterotrophic predators, actively hunting down and predating on other smaller organisms. It's a total Jekyll and Hyde situation. So all of this raises a pretty dramatic question for organisms living in such chaotic microscopic environments. What actually happens when conditions turn unfavorable? Let's explore the incredible survival mechanism of our next group. Section 4. Slime molds, the decomposers, the saprophytic survivors. Okay, picture this. During suitable conditions, these saprophytic protists spend their happy days slowly creeping along decaying twigs and leaves, just engulfing organic material. They form this giant spreading aggregation called a plasmodium, which can actually grow and spread over several feet. But when unfavorable conditions inevitably strike, that massive plasmodium changes gears. It differentiates and forms these fruiting bodies that bear spores right at their tips. Now here is the massive exam trick you absolutely must remember. While the main body of the slime mold lacks a cell wall entirely, their spores actually possess true walls. These true walls make the spores extremely resistant. We're talking survival for many, many years, even under severely adverse conditions, until they are finally dispersed by air currents to start the cycle all over again. Section 5. 4 types of protozoans, the heterotrophic predators and parasites. So, protozoans, these guys are widely believed to be the primitive relatives of animals. They live purely as heterotrophs, meaning they function exclusively as either predators or parasites. Now, examiners love to test you on exactly how these 4 major types of protozoans move, so make sure to match the structural hack to the specific name. First up, amoeboid protozoans, like amoeba. They move and capture prey by putting out pseudopodia, which literally translates to false feet. And fun fact, marine amoeboids often have silica shells on their surface. Next, flagellator protozoans. Just as the name implies, they utilize flagella to move around. Then we have ciliated protozoans, like paramecium. These are highly active aquatic organisms that coordinate thousands of tiny cilia to steer water, which is laden with food, directly into a cavity called a gullet. Finally, we have sporozoans. This is a diverse group, but they are categorized by having a highly infectious, spore-like stage in their life cycle. It's super important to ground these protozoans in their real-world impacts, particularly those parasitic forms. You really need to memorize 2 specific real-world diseases tied to these guys. First, a flagellated protozoan called trypanosoma is the culprit behind sleeping sickness. Meanwhile, the most notorious sporozoan out there is Plasmodium, the malarial parasite. It causes malaria and continues to have a staggering, devastating effect on human populations worldwide. Section 6. Key exam takeaways. The ultimate comparison. All right, the most crucial point of the explainer is coming up right now as we move into our high-yield revision summary for the entire Kingdom Protista. Let's pull all these sneaky classification differences together. This breakdown is your ultimate screenshot-worthy guide for your test. It cleanly separates the most commonly confused structural boundaries of these organisms. Remember, chrysophytes, indestructible silica shells fitting together like a soapbox. Dinoflagellates, stiff cellulose plates. Euglenoids, they ditch the wall completely for a flexible, protein-rich pellicle. And slime molds, no wall on their main plasmodium body, but they do possess highly resistant true walls on their spores. Memorize these specific comparisons, and I promise you, you will effortlessly navigate even the trickiest taxonomy questions on test day. I want to leave you with this final thought. Today, we've systematically organized a kingdom containing organisms with indestructible silica armor, shape-shifting autotrophs that literally become heterotrophic predators in the dark, and protozoans that steer their food with thousands of tiny coordinated hairs. If all of this wild diversity is categorized under just one single kingdom, what other bizarre combinations are hiding just out of sight in the microscopic world? I hope this explainer leaves you thoroughly prepared to absolutely ace your exam and maybe even a little eager to keep exploring the microscopic wonders of biology. Keep studying, stay focused, and I'll catch you in the next explainer.